So for today, welcome Pablo Paioni. Pablo Paioni is a clinician from the Children's Hospital in Zurich, and I'm very happy that he will present to us his infrastructure development project, which is called SwissPKcdw, which actually aims on building up a clinical data warehouse for doing pharmacokinetic modeling. With that, I give the floor to Pablo.
Thank you, Sabine, for the kind introduction, and thank you for inviting us to present our SPHN infrastructure project. I will be presenting on behalf of the whole consortium, and I will tell you in the end who everyone is. So as you can see from the title, our project is interested in optimizing pediatric dosage regimens, and we will want to do that based on data in a clinical data warehouse, which we use for pharmacokinetic modeling. So let me show you a little overview over the other SPHN projects that have been funded 2017 and 2018. There are a total of 13 infrastructure development projects and 11 driver projects. And you can see that our project is here. It is a little bit in between biomedical platforms and patient oriented projects, which I think is actually a very good definition of what we would like to do and what we are doing. So let me start with a little bit of background on what we do, and then I will go on showing you how we have been doing it and for what purpose. And also I will then show you some examples of the challenges that we encountered during our project so far, and in the end then present you with the actual situation, where do we stand and where we are going. So why are we doing this? We are interested in pediatric dosage recommendations, and we are doing this project mainly because of the actual situation regarding pediatric drug usage. So for practical and economic reasons, clinical trials to find the ideal dosage regimens have generally been restricted to adults in the past, and were not followed by trials in children. And this is the reality, and this situation leads to the fact that we are left with drugs that are used in adults and have recommendations for dosage in adults, and we have to use them in children because we need them, but we don't have a precise recommendations about the dosage. So we use those drugs so-called off-label, which means that the drug is actually licensed in the country, so in Switzerland, but also in other countries, but it's licensed for adults. And so if you look into the leaflet, when you open the box of the drug, then it might say it's not, it should not be used in children below a certain age. So this situation is clearly not ideal, and it increases the risk for both severe adverse events due to overdosing, but also for treatment failure due to too low doses. So why is that so difficult to find the ideal dosage in children? Why is it not possible to just adapt the dosage from adults to the child? So actually it might sound easy. So you have an adult who is which is 15 kilograms of weight, and you use 100 milligrams of the drug, and then you want to apply it to a child who is half this weight. So you spontaneously would use half the quantity of the drug. And the problem is that it's not so easy, that the human body doesn't work that way, especially in growing children. And there is a certain number of physiological parameters, as you can see here on the left of this slide, that have an influence on the metabolism of the drug once it is taken or injected. And these physiological parameters, which are, for example, the intestinal absorption through the gut, or the hepatic clearance through the liver, or renal clearance to the kidneys, or even the volume of distribution in the body, these parameters, they don't stay the same throughout life, but they change over time, especially in the period from the newborn until about 12 years of age. So they can vary, and you can have both situations. You can have one enzyme, one parameter that increases its activity, but you can also have the opposite. So one enzyme that decreases its activity over time. So depending on which drug you're using, you're considering, these different parameters will play more or less important role in the metabolism. And that makes it difficult to really adapt the dosage in children. So it can be that the child needs more of a drug related to the body weight than an adult because of that. So there is no common rule to predict the ideal dosage in children, and you cannot just adapt it very easily from adults. But at the moment, that's the way it's done for those drugs that are used off-label. And this situation leads to a high heterogeneity in dosage recommendations for children. So even within a small country like we are in, every hospital might use a different dosage recommendation for a certain drug. And this is something that it was recognized not only by us during this project, but also by politics. And that's why the Therapeutic Product Act was revised 2018, allowing now the Federal Council to allow a collection and harmonization and evaluation of data relating the prescription of medicinal products in children. And so the Confederation arranged for a database to be established with the goal of harmonizing dosage regimens in children, and this task was taken up by SwissPKcdw. SwissPKcdw is an initiative from all 8 Category A children's hospitals in Switzerland, and is led by Christoph Berger, who is also the PI of this SPHN project that we are doing. And SwissPKcdw has the goal of get rid of this heterogeneity in Switzerland and harmonize the dosage for children. And the way they do it is through an harmonization process. So they developed a very smart way to harmonize those regimens, where they first ask the children's hospitals for a certain drug. So there will be their data experts, which are usually pharmacists from the hospital. They will provide the dose that it's used in that particular hospital and send it back to the expert at SwissPKcdw. And there will be first a literature review, and then in the end a proposal of a harmonized dosage, which will be then go into the harmonization tool and go back to the hospitals where other experts, usually doctors, using those drugs, and depending on which drug is taken under consideration, it will be a general pediatrician or an infectiologist or an neonatologist, and those experts will look at the proposal and then decide if they agree with that, based also on the data that from the literature that will be sent together with the proposal. And then there will be either a consensus if all 8 experts agrees, and if there is a consensus, then there is a national harmonized dosage proposal which goes back to the hospital. And if there is a dissent, so if the experts cannot agree, then there will be another round with a new proposal. But if this doesn't work, and mainly the main reason usually why this doesn't work is that there is a lack of data from the literature about that drug. And if that doesn't work, so we came up with the idea of our project, SwissPKcdw, where we want to collect routine data, as many as possible, about drugs that we use in children and about drug levels that we measure routinely in the clinic, in order to have a data set that we can use for modeling and for finding the optimal dosage for a certain drug. So that was a starting point, a little bit of our project. So the main goals of SwissPKcdw are, in the end, to develop this infrastructure, so this Swiss pharmacokinetics clinical data warehouse, and an online platform called SwissPKcdw for pediatric pharmacokinetic modeling. And on this platform, we would like to, or we are developing and providing tools based on open source software for pharmacokinetic modeling in order to predict the ideal dosage regimens for children based on the data that are collected in the CDV. And then we also want to make these modeling tools and this knowledge and this data available to the expert community via the SPHN network. So we want in the end to build a national modeling platform to share data, knowledge, and expertise. So we did that. So we applied for the funding in 2018, and we decided to build this infrastructure. And we were aware from the beginning that, that in order to build the infrastructure, you also have to test it. So we identified 3 drugs that we use often enough in the clinic, and for which we also do routine drug level measurements. And we decided to build the infrastructure, testing it with data about those 3 drugs. And those drugs are 2 immunosuppressant drugs, cyclosporine and tacrolimus, and then an antibiotics, gentamicin, which is very commonly used in children. Then we also decided to include pharmacogenetic information and analysis in the project, because we are aware that pharmacogenetic will play an essential role in the future of personalized medicine. So we designed SwissPKcdw to allow integration of pharmacogenomics data, and we are planning to assess the influence of genetic variances on the metabolism of those 2 immunosuppressants drug by performing genetic analysis, especially on this enzyme here that is central in the metabolism of these drugs. And then we will retransfer the results into our platform and into the CDV, and use that in the modeling. So this is mainly how the whole project looks like. In this figure, you can see in blue the data provider, which are 2 children's hospitals, the University Children's Hospital in Zurich and the University Children's Hospital in Basel. Then you have the University of Basel, where the genetic analysis will be done in the lab of Professor Henriette Meyer-zu-Schwabedissen. And we will send bio samples in a coded way to Basel, where the genetic analysis will be performed, and then the results will be transferred back via the Biomed IT node in Basel into our platform. Here in gray is the LeonardMed, the Biomed IT node in Zurich, which is the platform where our tenant will be stored, and we will be able to transfer those data back there and include them into the clinical data warehouse. And I'm going to show you later in the slides how we will do that a little bit more in detail. Maybe just quickly again about the University Children's Hospital in Zurich. We have built a data lake where we export all the patients' data from the clinical informatics system, and we are able then there to process them and to prepare them for the transfer. Then we will have users, as you can see here on the right side. We will have authorized users with different clearance levels that will be able to either use the data to do their own modeling or to just explore the data that are already in the CDV or see results of already written modeling scripts in order to have a good idea of what's there. So now I'm going to walk you through the main challenges that we encountered during the project, mainly in different areas of the project, and then by doing that I will then also highlight then how we what we did to overcome these issues and how we designed our infrastructure. So there are many areas where we encountered problems or challenges. One area are the data, and it's about harmonization and safety and privacy, as you all are aware. During a SPHN project, those are our main challenges. Then we have the IT area, and I will show you a little bit more in details what the challenges here were. Then we have regulatory issues within the SPHN with legal aspects and project classification. And then in the end, we also have challenges related to the collaboration, because it's clear that if you have many players in a project, then communication and the way you coordinate is really important for the success of the project. So let's start with the challenges in the data. So one main topic is harmonization, and that's something that is within SPHN, and it's one of the goals also of the SPHN initiative, is to make possible that data from different sources from 3 different hospitals can be used together to answer a scientific question. And in order to do that, you first have to process those data in a way that you can compare them and that you can use them together. Because for those of you that worked in a hospital before, it becomes very quick clear that every hospital has its own system, informatics system, and its own way to collect data and generate data, and that makes it difficult when you want to use those data then for a scientific project. So one example of the issues that we had during our project was, for example, the issue with the LOINC codes. So the major challenge was the fact that the LOINC codes are not currently used at our hospital in the lab. So we first had to define the LOINC codes for our analysis together with our lab. We received, of course, support also from the SPHN community, and this was feasible. And the second problem was that the SPHN core data set was suggesting to use the L4CH lab concept for the LOINC codes, but required to use only one code. But in fact, in the L4CH lab concept, there is not only the LOINC code, but you also have 3 additional pieces of information about the manufacturer, the kit, and the instrument used for a certain analysis, which are very important information if you want to analyze the data or describe your methods. So we had to clarify with the semantic group at SPHN what to do, and we were advised to only use the LOINC code from the L4CH lab concept. And we did that, but by doing that, we thought that those three additional information would be lost and wouldn't be there. And those are important, as I said, for example, when you need to describe your methods and maybe compare different labs doing the same analysis. So we went to Professor Lovis from the L4CH lab with an idea of linking the unique identifier number of the lab to the L4CH lab concept. So in order to basically know which lab made the analysis and to be able to track back also the information about manufacturer kit and instrument used. And so we did that and it was successful, and L4CH lab agreed to link the UID in their concept, and so we could overcome this problem. Then another problem regarding the harmonization was the SNOMED CT coding for the route of administration of a drug. And it became clear while looking at our data that we had route of administrations that are not yet coded in SNOMED. So we had to request to the SPHN semantic group to add this specific route of administration to the code. Then we had challenges about safety and privacy. One example is the birth date of a patient, and maybe you're not aware of these, but in pediatrics, when you're doing research, very often it's really important to know the exact age of a patient. So 2 or 3 months different, they can have a big influence depending on the analysis that you're doing. So we wanted to have the exact age of the child, but we were not allowed to export the birthday. So we came up with this solution to generate an offset for any date time exported in our data, so that we will export the date of birth, but it will be modified with an offset so that not the actual date of birth is showed, but all the other dates of the administration of the drug or the date of the analysis will also be modified according to the same offset, so that the differences between will remain the same, and we will be able to calculate the exact age of the patient at each time point. Then we had also, regarding safety and privacy, the results of the genetical analysis, and we first wanted to bring those results back to the hospital within our data lake, but we then decided that this was too critical, and we didn't want to put those results together with the patient's data in the hospital, and so we decided to transfer them directly to our tenant on Leonard Med through the Biomed IT node in Basel. Another challenge, which is more a general challenge that every SPHN project has, is about data quality. And the problem is that we are using routine data. And those are data that are generated during clinical routine management of the patients and are collected. So the requisites to make research out of those data is to improve the quality of those data away from a retrospective data quality level, more toward a prospective data quality level. And this is only possible through a chain of several processes during the data export, like harmonization and extensive data check, and in order to improve the quality of the data. And these processes are mainly or very often IT based. So I'm going to continue with the challenges regarding the IT part of the project. So as I said in one of the first slides, we generated a data lake within our hospital IT system in order to export all the patients' related data and to be able to process them in a separate way within this sensitive data lake. And this is something that we, from the beginning, we realized is really a pioneer project. It needs a lot of know-how and manpower. You have to link incompatible source systems. You have to make sure that you can keep track of the changes. So it's not easy done. So we decided from the beginning to outsource this data lake implementation to an external company. And that was a really good decision now looking back, because they were able to do a great job and build a data lake that is really helping toward the goal of our project. Then we had the problem that semantic requirements from the SPHN semantic group were not always routinely available in our source system. So we had to create mapping systems to link attributes and coding to the data. For example, the drug that we are looking at, the drug administered, would have to be linked to the ATC code. And in order to do that, you have first to map the drugs that are extracted and used, and then link automatically the ATC code to that data set. Then we needed also new tools in order to fulfill the requirements and make the project work. New tools, for example, for coding the data, for the pseudonymization of the data, tools for the encryption and decryption. So we also, of course, had a lot of support from the scientific IT services, from the ETH, in order to do that. And we generated, for example, our own concepts such as the pseudonymization algorithm in order to be able to process the data and transfer them securely. Then there were some delays with the IT infrastructure. Lately, there was a downtime of Leonard Med due to possible cyber attacks. Those are things that you cannot foresee, and also that no one can change or have an influence on. So it's something that you have to take into account when you're doing such a project. And we tried to keep a very tight schedule and milestones throughout the project, so that we were able to quickly react to situations like these and minimize the delays. So here this is a data flow within the children's hospital. It's how it's done in our project. So you have at the bottom, you have the different source system where the patient's data are generated and stored. So you have the admin data, then you have the clinical information system, the lab information system, then you have another different clinical information system used in certain clinics within the hospital. And out of those source systems, we exported the data into this data lake that I mentioned before, in order to be able to process the data within the data lake in a secure way. And in the data lake, we had to apply the criteria to the data, because we wanted to have a look at those drugs that I mentioned in the beginning. So we didn't want all the data in our system. We wanted to filter them, and we applied those 4 criteria that you see here. So first of all, only patients that agreed to our general consent. So they agreed to the use of their data or the data of their children for research purposes. Then we clearly filtered for the drugs that we were interested in. And then we decided also, in order to increase the quality, as I mentioned before, to focus only on inpatients, because the prescription system for inpatients is much more precise and allows to extract data with a higher quality. So we focused on those patients, and we are leaving out at the moment outpatients. And also we decided to focus on patients or prescriptions that were made through a new tool that was introduced in our hospital last year, an e-medication tool that also helps to increase the quality of the data that are extracted. So then the data and the files will be encrypted and transferred to our project space on LeonardMed, and I will show you later what happens there. We defined an export structure of the data. You can see here the different domains. So the patient, we have information about the patient, about the diagnosis, then the lab results, about the medication, and then we have information about the weight and height of the patient. And this data structure has to be then transferred to our CDW, to our clinical data warehouse, which is built with a software called Optimised. And in order to do that, we had to develop a data model to define the structure of the CDW and to be able to import the data. So the requirements of this import workflow were the fact that the data should be automated imported. So we are now dealing for this infrastructure project. We are not dealing with huge data set because we are still in the phase of building the infrastructure, but the goal in the future is to collect as many data as possible. So it has to be an automated process. There is no point of having to import then the data manually. And then the second thing is that as I mentioned, we are exporting first our data into this data lake, and this export is being done regularly. So every time everything is exported and is transferred according to the criteria that I mentioned. And then what we want to do on the other side in our CDW is to take up only the differences, so only what has changed or new data, and to leave what is already there unchanged. So it has to be an incremental update of the Optimised database. Then we have to implement data checks in order to improve the quality of the data. And if we find error, we have to generate standardized error reports. And then we also have to find a way to handle changes in general consent, which means that if a patient decides that he doesn't agree anymore with the use of his data for research, then we have to be able to identify the patient in the data model, in the clinical data warehouse, and to delete his data there. The data check there are different kinds of data check, as you can see here, but I'm not going to go into details about that. So this is how it's done. So we basically get the CSV files from the transfer from the hospital, and then they will be first on the tenant in a temporary way. The first data check will be performed to find problems or errors, and then the data that came in will be compared to what's already there in the permanent data set, and in order to find the differences, so that we are able to only import the changes into the Optimised. And also there will be data checks and problem reports, and then the final version will be then made permanent, so that when the next data set comes in, this process can be done again. So I'm going to continue with the challenges about mainly regulatory issues within the SPHN. So there are legal aspects. One example and one main challenge that we had lately and during our project is this data transfer and user agreement. So it's something that clearly it's needed to assure that everyone is dealing with the data in a correct and secure way. And it's something that we also knew from the beginning. It's not easy to get all the signatures, and so we actually started quite motivated and tried to bring that forward. We had a template from the SPHN, which was great. We adapted to our project, and then we were quite happy when we could get the signature. So the first, the DTUA was signed by the data providers, which are the hospitals, and then the data recipients, which is the project leader. And we were quite happy when we managed to do that. But then later on during the project, it was decided that this wasn't enough. So not only the data provider and the data recipient has to sign this agreement, but also the processors of the data. So the biomed IT nodes must be included. So a new template was generated by the SPHN, and we are now in the process of getting the signatures of all the involved players. And this is something that takes time, and it's a requisite in order to be able to transfer data. Otherwise, without that, you cannot transfer data. And if you cannot transfer the data, or the real data, you cannot properly test the infrastructure. Then we had an issue with the project classification. I didn't say that in the beginning, but we actually applied for a driver project, and because we thought that, yeah, we are building an infrastructure that then we want to really use extensively, and so we came up with different scientific questions, and we applied for a driver project, but were awarded only of an infrastructure project for 12 months, which is great, but the time schedule is much tighter. So this is an issue, and we had so we put in a request for a cost neutral prolongation until the end of the year, and we received that, but we are still focusing on finishing the project before that date. Then I mentioned in the beginning, collaborations are central, very important within this kind of projects, and they bring also challenges because you have different source systems in the different hospitals. You don't have everywhere the same manpower, so we had to build flexible IT structures, and we had to support, and we are supporting the local IT and data management support. But I have to say that we're really happy. We really found collaborators that are doing great work and are helping to bring this project forward. And then communication is also an issue. When you have different partners, we had 16 interdisciplinary partners and 7 institutions involved. So we established regular project meetings and documented what was discussed. So we were using GitLab for repositories, and also the project coordinator plays a central role in order to guarantee the success of the project. So now I'm finished with the challenges, and I want to quickly show you where we are now. So our SwissPKcdw is running toward the finish line, but as you probably, many of you will know, such a project, you don't have one finish line, but you have several ones. So we are now focusing on the high priority deliverables while we are shaping the end product of our infrastructure project. And we are aware that there is always room for improvement, and it's good if you realize that during your project and you can improve and make it better. Sometimes you have to put some of the ideas aside for a moment to be able to finish with the main goals. And then even though we have this extension until the end of the year, we are still aiming to finish the core infrastructure until the end of August. Sorry. So this is how the end product looks like in terms of data transfer. Some of these you will recognize from the previous slides. So this is the source systems at the hospital, transferred into the data lake where they will be processed and harmonized, and then they will be sent to through the Biomed IT node in a coded way and encrypted way to be transferred to our tenant on Leonard Med up here. The same will happen from the University Children's Hospital in Basel. The data will go through Psycore, which is the Biomed IT node in Basel. And then what we are also doing is regarding the genetic analysis is providing samples from the University Children's Hospital in Zurich. So we are doing regular queries to identify patients that came in recently for a blood test so that we are able to take leftover of this blood and send them to Basel for the genetic polymorphism analysis. And those data will be also then transferred into our tenant through Psycore in Basel. So now just quickly a little bit more into details, what happens on the other side, on the tenant side. As I said, the data from the Children's Hospital in Zurich, they go through the data lake, then they are exported and sent as CSV files. Then we have this import workflow I talked about earlier. We have problem reports, and then the current data will be compared to the new data, and only the difference will be imported into the Optimised, and then from Optimised the data will be split into spaces. For each drug, one space. Over the time of the project, we also included voriconazole, which is an antifungal drug which we use in pediatrics, and for which we measure also regularly drug levels. And from Optimised, those data will be exported again in CSV files, and then will be fed into an R script, and within R, the modeling will be done and created in order to, in the end, to find the optimal dosage for a certain drug. And what we are also doing is a web app. So this is a web-based interface in order to be able to navigate through the data that we have in the clinical data warehouse. This allows also people with no modeling skills to look at the data. They can select, they will be able to select the drug of interest, then they will be able to select some parameters of the patients, and then they will be able to generate reports to see how many patients, data of how many patients do we have, what age have the patients, and so on. And they will also be able to generate plots to look at the data in a graphical way, and also to look at modeling results from scripts that are running in the background. So that's the status at the moment. So we are actually we feel that we are on time, and we are running toward the finish line. And but after the end of the project, we want to continue. And what's the outlook beyond the funding period? We would like to extend the population pharmacokinetic modeling to as many drugs as possible in pediatrics, in order to optimize dosing of already used drugs in children, especially the off-label ones, to increase the number of licensed drugs for pediatric patients, and also to support the development of future new potential life-saving drugs for children. And it's clear that we can only do that by including the remaining pediatric hubs in Switzerland, represented within the SwissPKcdw initiative, and that's one of the main goals for the phase after the ending of funding period. So this is the consortium, and this is my last slide. I would like to thank everyone participating to the project. Christoph Berger at the University Children's Hospital in Zurich is the project leader. Then Vera Yegi is the data manager, and she's coordinating also the project. She's doing a great job, and we are very thankful for all what she did so far. And then we are collaborating also with the University Hospital in Zurich, with the ATH, of course, with the SES. And then at the Institute of Pharmaceutical Sciences, we have Professor Stephanie Kramer, who is taking care of the modeling scripts. Then we have the University Children's Hospital in Basel, also as data provider. We are collaborating with SwissPKcdw in the sense that we will then extend our project to the other clinical pediatric hubs in Switzerland. And then we have the University of Basel, as I mentioned, doing our the genetic polymarkers analysis. And with that, I'm finished, and I thank you for your attention.